part of a flower which is a male reproductive part, and it is attached to a stalk like filament which carries anther in its tip, which is called as stamen. Okay, we perform this culture to obtain the haploid plants from naturally diploid plants. So haploid plants means those having the half of the chromosome number, means the letter n, we do the small letter n we donate for the chromosome number for the haploid plants, and for the diploid plants we use the term 2n. So those plants which are having a represented by single letter, small letter n, are called as the haploid plants, means having half of the chromosome. These are originate from the gametes or the gamete like cells that do not go through the fertilization. Okay, this have single set of chromosomes. Now what are chromosomes? These chromosomes are the thread like structure located inside the nucleus in the plant cell. Okay, so this, what is this anther culture? It is a procedure, okay, for obtaining the haploid plants from the diploid one. Okay, now this, it is used for obtaining, okay, in plant breeding it is used for obtaining the haploid plants, and by what means? To by crossing the between the species with the different chromosome numbers. Okay, now here you can see below in the slide there is one figure which is showing the procedure of this anther culture. Okay, what you can see here, that yellow portion, this anther is bilobed. Okay, bilobed it is, and it is attached to this stalk like structure, that is stamen. It is inside the flower. So what you have to do is, you have to separate it from the flower and you have to put it in that culture, and after some time that some shoots will develop called as the new haploid shoots. Then the objective, okay, the primary objective of this anther culture technique is, I have told you, to obtain the haploid plants, okay, by using the regeneration of the totipotency, okay, of the anther. So this totipotency means the capacity of the plant cell to develop into an entire plant. Okay, so there are two methods in anther culture. The first one is the embryogenesis and the second one is the organogenesis. So the embryogenesis is a method in which the anther behaves as a zygote and forms embryoid. Now what is this zygote? So this zygote is a cell formed from the union of the two haploid cells. Okay, it is a cell which is having a single set of chromosomes, and it is a cell which is formed from the union of the two haploid cells. That's it, and it forms the embryoid. So what are these embryoid? These are the mass of the plant tissues, okay, that resembles an embryo and which give rise to a haploid plantlet. Now this organogenesis. In this method, the anther undergoes the cell division repeatedly to form the callus tissue. What is this callus tissue? Callus means the unorganized growth or the unorganized mass of the cells. So the callus tissue it forms, which later give rise to the haploid plantlets. Now here in this slide you can see the procedure given. In the previous slide you have already seen an introductory part of the process. Here you can see it in a more advanced form. See here the selection of the bud from a plant has been done, and then it has been capped, and after that it is kept in the laminar airflow chamber. After that the surface sterilization is done, then distilled water is added for washing, then it separately it is capped, then cutting of the bud takes place, then its removal, then the removal of this stamen from the bud takes place. The stamen, I have told you, the stalk like structure which is having the anther bilobed structure at its tip. Okay, then we will separate this filaments, means that stamen, from the anther, and we will keep that anther in the culture media which is having anther culture nutrient media which we prepared in the laboratory. Then obviously, as I have told you, that there are two methods. First one is embryogenesis and the organogenesis. So the both methods have been shown here. See, the organogenesis is here and embryogenesis is here. Okay, so the callus develop in organogenesis, and after some time haploid plantlets you can see from the callus, and here the growth you can see in that anther, and then haploid plantlet grows. Then the factors which influence this procedure, the anther culture, is the intrinsic factors and the extrinsic factors. So the intrinsic factors include the anther wall, which is a provides the nourishment to the developing stages of the anther in this procedure. Then the stage of the anther, there are three stages are there: pre-meteoric, meteoric, and the post meteoric stages. Okay, then the pre-meteoric is the stage when the first meteoric division occurs in the anther and where the pollens are immature stage. In the second stage there is division of the pollen, and in the third stage there is the development in the pollen grains occurs to form the embryos. Then bird size, here it up to 17 to 22 mm in length, and age of the plants, so here the younger plants are preferred. Now these extrinsic factors are the culture medium, the temperature, the role of the activated charcoal, and the pre-treatment method. So in this culture medium, like sucrose, iron, these are very important growth regulators like auxin, cytokinin. It plays an important role to form the haploid plants from the diploid one naturally. Then temperature, then so it varies with the different species, as in the example you can see of the dhatura. Okay, then role of activated charcoal, it plays a vital role in the removal of the inhibitory substances like both exo and endogenous growth hormones from the culture medium by stimulating the growth of an anther. Now pre-treatment method. So for the anther culture, pre-treatment of the bird is also suitable for the production of embryos. Okay, pre-treatment method, as example is given here, Nicotiana typicum. The bird is pre-treated, okay, pre-treated at a temperature of 5 degrees centigrade for 72 hours. Now applications, so it has various applications are there. Here I have listed 10 applications for you in this slide. You can you should go through once, okay, with this slide. Okay, then first one is like utility of this culture for the basic research. Okay, for basic research this culture has been used, and now also it's been in use. Then utility for the mutation purpose. What are mutations? The sudden irreversible changes in the genes. Okay, then used for the cryogenic studies. It is used to obtain the commercial variety economically. Important crop, rice, wheat, coconut, rubber plants, etc. are used to obtain the commercial variety of these plants. Then to produce the large number of plants in the short period. Okay, this anther culture is having a great role to produce the large number of plants in a short period of time. Then rapid generation of the homozygous genotypes after chromosome doubling. Then study of the genetic recombination in the higher plants. Study of the mode of the differentiation from the single cell to whole organism. Study of the factors controlling